Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I am Waqar Ahmad and today we will study about the biogenesis of Ats ribosomes. So this is the lecture number 4 about the ribosomes. Lecture 4. So in the previous lecture we have studied about the biogenesis of Ats, and today we will study about the Ats ribosomes synthesis. The Ats ribosome synthesis, this process is more complex as compared to Ats. It is more complex because many cellular regions are involved in the synthesis of Ats. Many cellular regions are involved in the synthesis of Ats. As we have studied that the Ats ribosome, it contains the subunits, the Ats. It contains 28s, ribosomal RNA subunit, 18s, the 5.8s and 5s. So these are the ribosomal subunits. These are the ribosomal subunits. The synthesis of ribosomes of eukaryotes, it occur in the nucleolus. It occur in the nucleolus. The nucleolus, it contain one of the region. Nucleolus contain nucleolar organizer region. That is called NOR. Nucleolar organizer region. The nucleolar organizer region, for example, this is the nucleus. Inside the nucleus there is present the nucleolar organizer region. This is the nucleolar organizer region. Now what the nucleolar organizer region contain? This region contain, the nucleolar region, it contain genes for it contain genes, or we can say it contain ribosomal DNA for ribosomes synthesis. For ribosome synthesis. All the 4 subunits of ribosome are not synthesized in the nucleolar organizer. The 3 subunits, the 28s, 18s and 5.8s, these 3 subunits, they are synthesized in nucleolar organizer, while the 5s subunit, it is synthesized in nucleoplasm. The gene for it, the genes for this subunits, they are present in the nucleoplasm, while the ribosomal proteins, they are synthesized in cytoplasm. While the mature ribosome synthesis occur, all the process complete in nucleolus. All the process complete in nucleolus. As the 3 ribosomal subunits synthesized in the nucleolar organizer region, the 5s subunit, it's synthesized in the nucleoplasm, while the outside from the nucleoplasm in the cytoplasm, the proteins are synthesized. So all the components which are needed for the ribosomes formation, so move to the nucleolus, and the mature ribosome synthesis occur in the nucleolus. Now there are the 3 main steps. The biogenesis of Ats ribosome, it involves the 3 main events. There are 3 main events involved for biogenesis. In biogenesis, the 3 main events are, the first one is the ribosomal RNA synthesis by nucleolar organizer, as I said earlier. Ribosomal synthesis by nucleolar organizer. The second one is the biosynthesis of ribosomal proteins. This is also one of the steps. Biosynthesis of ribosomal proteins. And the third one is the 5S ribosomal RNA in the nucleoplasm. So these three are the main events in the biogenesis of ribosomal RNA, the ribosome synthesis. Now the ribosomal RNA synthesis inside the nucleolus, the first one. The ribosomal RNA synthesis in the nucleolus. The ribosomal RNA synthesis in nucleolus. So first of all, it was studied that the nucleolar organizers involved in the ribosomal RNA synthesis. It was studied in South African frog. In South African frog. And the name of this frog was Xenopus laevis. Xenopus laevis. So in Xenopus laevis it was studied. This Xenopus laevis, it was mutant. It was mutant in nucleolar organizer region. Some genes were missing in the nucleolar organizer region. So the Xenopus laevis was unable to synthesize 18S, 23S and 5.8S ribosomal RNA subunits, while the Xenopus laevis was able to synthesize the 5S ribosomal RNA subunit, but these three subunits were not synthesized. This is because of the mutation in the nucleolar organizer region. The ribosomal RNA genes. The ribosomal RNA genes. The ribosomal RNA genes. The ribosomal RNA genes, they are present in the nucleolar organism. The organism contain many genes. Organisms contain many ribosomal RNA genes. For example, it was studied that the Xenopus laevis, it contains 450 ribosomal RNA genes, and these genes are present in tandem repeated manner. The genes are present in tandem repeated manner, or we can say in tandem repeats. So the genes are present in tandem repeats, and the genes are separated by spacer DNA. Separated by spacer DNA. Now what is spacer DNA? The spacer DNA, this is the non-coding sequences of DNA which are present between genes. For example, this is the gene of ribosomal RNA. This is also the gene of ribosomal RNA. This is also the gene of ribosomal RNA. So in the mid region between the genes, there are present spacer DNA. Red color, as I mentioned, so this one is the spacer DNA. So the spacer DNA, this is the conserved region that is present between the genes. This is also the spacer DNA. So the spacer DNA is the non-coding region of DNA which do not code any protein, but it separates the genes from each other. Now the spacer DNA, what is the function? The spacer DNA, it acts as a binding site for many transcription factors. Its function is that it acts as binding site for transcription factors. For transcription factors. When the transcription occurs, so the transcription factors come and it binds to the spacer DNA. Now the genes of ribosomal DNA, how they are present? They are present in such a manner. The ribosomal RNA unit is synthesized 1 unit. As a whole, 1 unit is synthesized. So how this 1 unit is synthesized? So this is the from ribosomal genes. From ribosomal genes, 145S ribosomal RNA unit is synthesized. Now further processing of this 45S occur. Further processing of 45S occurs and the mature subunits will be synthesized. So how it is done? So this process occurs in such a way. From 5 prime to 3 prime. This is the 5 prime region. This is the 3 prime region. To the 5 prime region, this region is considered, this is the spacer DNA. Next, there is a region of 18S ribosomal RNA. This one is 18S ribosomal RNA. This is 18S. After 18S, again there is spacer DNA. Then next is 5.8S. This is 5.8S. Then again there is spacer DNA. And then to the 3 prime region there is 28S ribosomal subunit. So this is as a whole, this is 45S ribosomal RNA unit is synthesized. This one 45S. So in such a way it is synthesized from ribosomal RNA genes in such a way. Now these, the red color as I have mentioned, so these are the, so this is the spacer DNA. This is spacer DNA. This is also the spacer too. So these spacer DNA, they are present, the spacer, now this is the RNA. So this spacer region is present between the subunits, the 18S, the 5.8S and the 28S. So when the maturation occurs, so how they are matured, the 18S, these spacer regions will be cleaved. The 18S, when it goes to maturation, so it is methylated. During transcription, it is methylated rapidly. So it has been studied that 18S 46 methylations occur on 18S. 46 methylations occur on 18S and 71 methylations occur on 21S methylations. The 28S and the 18S are the more conserved, so they are more methylated. So the regions, the 18S, the 5.8S, so they are the 18S subunit. Then the next is the 5.8S subunit, and then the next is the 28S subunit. In the nucleolar organizer region, so in this region, these are the loops of nucleolar organizer. So the nucleolar organizer, here the synthesis of the ribosomal RNA subunits occur. The synthesis occur. So here the subunits are synthesized. For example, the 18S, also the 5.8S and the 28S. So these subunits, it come to the next region of nucleolar organizer. This is the nucleoplasm. So this is the next region. This is called the dense fibrillar region. So this dense fibrillar region, the subunits come here, and then the next is the 5S ribosomal RNA. The 5S ribosomal RNA, it also come to this region. The 5S also come to this region. The 5S come, and also the proteins from cytoplasm. So this is outside the cytoplasm. The proteins also come here. So here the synthesis of the ribosomal subunit starts, and in the last region the maturation occur. In this region, maturation occur. So first of all, the 40S subunit is synthesized. First, the 40S subunit is synthesized. In the 40S subunit, it moves first of all to the cytoplasm. So this will be the 40S subunit move first, and then the 60S subunit, it take about 70 minutes more. The 60S, it also move out, but 70 minutes later from 40S. In the synthesis of ribosomal RNA subunits, the RNA polymerase 1 is involved. The RNA polymerase 1, it is involved in the synthesis of ribosomal RNA subunits. Synthesis. And also some transcription factors. For example, in the synthesis of these three subunits, transcription factor 1. The transcription factor 1 means that it activates the process of the transcription. While in the synthesis of the 5S ribosomal RNA, the transcription factor 3A is involved. In 5S, the transcription factor 3A is involved, and here the transcription factor 1. So in this manner, the synthesis of ribosomes, it occur in the nucleolus. So the fully maturation of ribosomes, it completes in the nucleolus, in this part of the nucleolus region, and then the ribosomes, they move out into the cytoplasm. Now the proteins which are involved in the ribosomes, so the proteins also synthesized in the cytoplasm, and these proteins, it move to the nucleolus, to the second part of nucleolus. This is the fibrillar center. So this is the fibrillar center. In the fibrillar center, the loops of the nucleolar organizer are present. Next is the dense fibrillar component. This one is the dense fibrillar component, and the last one is this one is called the granular component. This is the granular component. So these three are the parts of the nucleolus. So in the fibrillar center, the ribosomal DNA, the genes are present. Here the subunits are synthesized. In the dense fibrillar center, all the components move, the process is started, and in the granular component, the maturation of ribosomes complete in this part of the nucleolus. So this is about the synthesis of ATS or ribosomes, and this is today's lectures. Thanks.